So I think we're good. It's time. So, hi everyone. My name is Zeeshan Ahmed, and I am assistant professor at Rutgers University. And I represent my lab, and we are part of Rutgers Institute for Health and Robert Wood Johnson Med School, Department of Medicine. So, describing some. First of all, I welcome you all to join this interesting workshop, which is about IntelliGenes and two students who are going to present the complete workshop include William and Rishabh. They both are part of the lab and both are the students at Rutgers. So, describing some skills and research interests of our lab, we are mainly focused on the development of artificial intelligence and machine learning techniques and utilizing machine learning techniques to analyze multiomics and clinical data to predict different diseases and identify novel biomarkers. So our contribution towards our goals include IntelliGenes, which is an AI ML pipeline for biomarker discovery and predictive analysis. During this workshop, we will be mainly covering two different products. One is command line version of the IntelliGenes, which William will describe along with the research objectives and test cases, and then Rishabh will explain and guide you through the graphical user interface of the application, and then we'll open the house for your questions if there are any and guide you how to use it. There is on the right side, hopefully, there's a chat or question answer option. Please feel free to put your comments or questions over there. Any of us will definitely be able to reply that. So, William, please take from here, and yeah.
Thank you for the introduction. As said, I am Will Degroat. I am a research scientist at the Rutgers Institute for Health in Dr. Zeeshan Ahmed's lab. And today I'm going to be talking to you about IntelliGenes, which is the artificial intelligence and machine learning pipeline for biomarker discovery and predictive analysis. The second half of this presentation will be given by Rishabh, who is my colleague in the lab. Rishabh will be walking you through the IntelliGenes graphical user interface. We'll be performing a demonstration, and if you would like to follow along, we highly encourage it. We're here to answer your questions throughout this workshop. If you do have any questions throughout the presentation, feel free to put them in chat. We will see them. We'll answer them right away. It's a lot easier if you ask your questions when you have them rather than waiting to the end of the presentation. So we don't have to circle back, and we can get your questions addressed right away so you're not confused through any part of the presentation. With that said, I'm going to start by giving us some background on IntelliGenes, why we decided to develop it, and what challenges that we think we have addressed in its creation. So with that said, I want to talk about
Will, just one quick thing. I think your microphone might have switched off automatically.
Oh, can you hear me now?
Perfect.
How much did I miss there?
Just like the last sentence.
Okay. So to try back a bit, we want to start first by going over two previous papers that predate IntelliGenes. These were both published in 2022. They were reviews where we asked a couple key questions for the development of our software. This first paper here, the Frontiers in Genetics paper, really centered around what precision medicine is. We were interested in learning how AI and ML techniques were applied in the field of healthcare and how they were improving outcomes. In this second paper, we delve more into the technical aspect. What techniques in machine learning and artificial intelligence were being implemented, how were they being implemented, and what could we do to address some of the challenges that the field was facing. So let's start off with that first paper. So what is precision medicine? Precision medicine is really the intersection of three different fields. I know in the figure there's four here, but I'm going to combine two of them for simplicity for today. Those fields are genomics, healthcare systems, and artificial intelligence. When we talk about genomics, we are talking about the genetic data of a specific patient. This can be in the form of whole genome or whole exome sequencing, where we're looking at the sequence itself, or RNA sequencing, when we're looking at the transcription of certain genes within the genome. These genetic factors give us an insight into the relationships between gene expression and disease. Healthcare systems encompass mainly electronic healthcare records. EHR records are really important, not only because they measure environmental factors that might contribute to a disease, but because they contextualize this genomic data that we have. When we're looking at patients, it's a lot more complicated than if we were looking at biological replicates per se, which some of my fellow traditional bioinformaticians in the presentation might be familiar with. These are a patient data. This is a set of data that is extremely diverse, and we need to be able to account for extraneous variables that might bias our examination by leading to different genetic profiles of patients. And now that we have these two data types, how do we interpret them? These data types are both wide-spanning. Genetic data can be terabytes and terabytes of data. The same goes for electronic health records. We need artificial intelligence and machine learning to be able to process this data and make sense of it. A human doing this would not be able to do it anywhere near as efficiently, maybe even at all, compared to the computer systems that we have in place. With these AI systems, we can measure the importance of certain variables, we can make predictions about diseases, and most importantly, we can synthesize healthcare information and genomic data to make it work together. Now, to move into the genomic data more so, what are we looking at when we talk about genomics? And our lab is focused on two main types of data. This is gene expression data and variant data. Anyone who is familiar with precision medicine might be aware that RNA sequencing is the standard for precision medicine. Famous examples are the use of RNA sequencing to classify cancer subtypes. If you look here on this figure, you see that cancer is one of the most highly linked diseases to artificial intelligence and machine learning techniques. One of the challenges that these techniques face is that these data sets lack a comparability across different experiments and different samples. That's one of the things that we wanted to address as a challenge with IntelliGenes. When we look at the other side of this equation, this is genetic data or variant data. Single nucleotide polymorphisms, or SNPs, have been highly linked to different genetic diseases through GWAS studies. We know that these regions are extremely important to the early prediction of a disease. Okay, technical issue in the chat. Sorry about that. Single nucleotide polymorphisms offer insight into the non-coding genome, which RNA sequencing doesn't, and they have been extensively annotated. Single nucleotide polymorphisms are very well studied. Databases that link to GWAS and other EQTL sources have a variety of clinical data linked to them. You might be familiar with ClinVar, which is a prominent database of this type. Because of this, we can extract disease-relevant annotations directly from a single nucleotide polymorphism. With these two types of data, we examined which techniques were being applied and what results were being seen. What we observed is that there is really no consensus for the best practices across artificial intelligence and machine learning as applied to these two different data types. There are examples where this data is used and predictions are made, but those predictions aren't generalizable. There are examples where the models that were being used were not the best models for predicting a specific disease or condition, just because of the nature of the model itself. And we wanted to remedy these issues by creating an expansive toolkit for scientists who might not have the technique or computational skill to understand the intricacies of machine learning and artificial intelligence. So that brings us to IntelliGenes. So what is IntelliGenes? At its bare minimum, IntelliGenes is a machine learning-based pipeline for predicting disease-associated biomarkers for discovering disease-associated biomarkers and predicting disease. IntelliGenes is a platform that is built to be efficient, user-friendly, and of course, open source. That means that if there are modifications that a user wants to make, it's easy to do in IntelliGenes. It's easy to craft a pipeline that fits your needs exactly, even without prior coding knowledge. IntelliGenes integrates these data types that we talked about previously, that being genomics or multi-omics data and electronic healthcare records, directly in the pipeline. Additionally, IntelliGenes is useful for training single disease prediction models from data sets that researchers might not have, but not have the computational know-how to train the models. IntelliGenes was initially targeted at translational scientists and clinicians who might have a wealth of data, but no real way to translate that into a good predictive model. So before we talk about the architecture of IntelliGenes, I want to talk about the CIGT format. And the CIGT format stands for Clinically Integrated Transcriptomics and Genomics. If you are a bioinformatician familiar with RNA-seq data formatting, this might look somewhat familiar to you. Essentially, what we do is we extract the read counts or the TPM values from a program like RSeam or feature counts, and we pair that with clinical demographics or additional clinical data. The main three that we look to include in all of our experiments are sex, race, and age. These variables do a very good job at contextualizing genomic data. For instance, if you are doing a comparative study, you might not want to be looking at an older male patient in comparison to a young female patient because the genetic signatures are going to be such vastly different. Additionally, to these variables, there are two columns seen on the left. This is ID and type. What ID is just a unique marker for each patient, and type is a classification for your training data on a disease or a non-disease. So in this instance, case and control represent heart failure and patients without heart failure. That's a case study that we'll get into in just a bit. But for now, it's just important that we understand the format of this data. This data is directly able to be loaded into IntelliGenes or other AI ML software and get predictions or biomarkers from the format without any additional computational modification. This is a very simple format. It's meant to be easy to understand, even if you're looking at it. Yes, there's a lot of numbers, but you know what each of them means. You have columns that are all representing a single feature. When we get into these later columns, these ensemble IDs, these are all representative of genes or non-coding regions that are being transcribed. This format is extremely expandable and data efficient. Because of the way that we have constructed the data type, we are able to reduce the size of RNA-seq experiments drastically, which helps for researchers who might have limited hardware capabilities. If a system lacks storage or computational processing, the CIGT format addresses that. Now, let's look at the architecture of IntelliGenes. So this is a very bird's eye view type image of our software. We'll get into each of these parts specifically, but what's important to know is that this is the initial implementation of IntelliGenes. This is IntelliGenes 1.0. The IntelliGenes software is split into three modules. The first being a selection module. This is where we are discovering biomarkers that we think are associated with a condition or a disease. The second is classification, where we are training a model to predict that condition or disease. And the third is what we call the IGene score. The IGene score is essentially a way to ensemble multiple predictive algorithms together to create a single understandable index for different biomarkers of interest by performing a almost differential expression-like analysis. We'll be able to tell if the biomarker is important to predictions, if that biomarker is overrepresented or underrepresented in case or control samples, and how that biomarker ranks across different methods for predicting. If we see that a biomarker is scored very highly in multiple methodologies for predicting, let's say random forest and support vector machine, which we'll get into a bit later, we can probably infer that that biomarker is biologically relevant, and that's why it's capable of making good predictions. So with that said, let's get into this first module, which is selection. The selection module in IntelliGenes is a combination of classical statistics and machine learning techniques. This module is used to discover novel or unbiased disease-associated biomarkers, meaning that aside from your RNA-seq data, no other information needs to be given to any of these algorithms to make a prediction on if a biomarker is disease associated. We thought that combining AI ML methods with classical statistics would be a great way to breach between an audience that might not be exposed to machine learning techniques, but has a background in maybe statistical genomics, because these different algorithms are performing the same function. We thought it might be more interpretable to a wider audience. Currently, IntelliGenes hosts a Pearson correlation, so a simple linear correlation between a variable and type, a chi-squared test, an ANOVA, and a recursive feature elimination procedure, which is based on a random forest classifier. Each of the initial 3 statistical tests is based on a p-value cutoff, while the recursive feature eliminator is based on a percentile cutoff. Essentially, it can be programmed to cut off, let's say, the top 10% of features selected in your data set. Once you have a list of biomarkers that you have selected, that is when you train a machine learning model on those biomarkers. These markers can be used essentially as a better prediction mechanism than a full data set because they're more targeted towards the condition we're looking at. There isn't additional information being lost to the model. Currently, IntelliGenes hosts 5 different classifiers. That is a random forest, a support vector machine, extreme gradient boosting, k-nearest neighbors, and a multi-layered perceptron, which is an artificial neural network. Each of these different classification algorithms serves a different purpose, which we'll get into. But the most important thing to note is that we provide an in-depth review of each of these classifiers' performance. If you look at this figure here, you'll see that for each classifier, we generate visualizations and metrics that can help the user understand what the software is doing in terms of which markers were the most useful for making predictions, how did the different machine learning algorithms compare to one another, and what might need to be tuned to create a better predictive algorithm. IntelliGenes also offers hyperparameter tuning within the platform. So let's talk a little bit more in depth about each of the algorithms and how we can ensemble them together. So the first 2 of note are random forest and XGBoost. These are both tree-based machine learning algorithms, but they both serve different purposes. Random forest is a very good out-of-the-box algorithm, meaning that it doesn't require extensive amounts of training data or extensive tuning on the back end to create good and useful predictions. ARF is kind of our recommended go-to classifier for anything related to precision medicine, just because it's so interpretable and easy to use. SVMs are great for single disease classification problems. Across literature, what we see is that SVMs usually perform, the best on these types of biological data sets. K-nearest neighbors is very useful for when we are dealing with 2 groups that are very dense. So 2 data points that you expect to be very distinct. K-nearest neighbors performed quite well in our trial for heart disease versus non-heart disease patients, just because these are such distinct groups. For an approach that might look at molecular subtyping, as in the cancer studies we discussed a bit earlier, k-nearest neighbors would not be applicable. We would want an algorithm that more mimics random forest or XGBoost. And then finally, the multi-layer perceptron or the artificial neural network. Artificial neural networks are the highest performing models, but they require a lot of data. For precision medicine studies, usually we're only dealing with samples in the ranges from 100 to 200 patients. Some of the big, really prominent cancer studies will get up to about 2,000 patients, but that's really the max you'll see. So these out-of-the-box, really efficient algorithms like random forest will be a bit better for the types of experiments we discuss here. The final part of this section that I want to talk about is this voting classifier. With IntelliGenes, we realized that in previous studies, most research groups were only using a single algorithm to make their predictions. What we found is that a 2 algorithm or a multi-algorithm approach, a multimodal approach, will perform much better as compared to using a single predictor. So we implemented a voting classifier that allows multiple predictors to be ensembled into one understandable accuracy score. This is the in typical trials, this is the most accurate classifier that IntelliGenes can produce. We recommend that when you're using IntelliGenes, you use a variety of different algorithms and ensemble them together to get the not only the most interpretable, but the highest accuracy predictions. Speaking of ensembling, let's go into this final section here, which is the IGene score. IGenes is built on SHAP, which is shapely additive explanations. SHAP is a way of measuring feature importance, so the importance of specific demographics or genes within the prediction of a disease. SHAP importances are generated for each of the classifiers in the IntelliGenes trial. So if you choose to use, let's say, RF and XGBoost, SHAP will generate importances for both of these classifiers. Afterwards, these datasets, these SHAP datasets are normalized, and we use a HHI index to compute the relative importance of different features in different classifiers. You might not be familiar with HHI. It is very similar to a Gini coefficient. We're measuring the level of inequality or entropy within a system. Finally, we generate an IGene score, which is an aggregated weighted importance of the different biomarkers, the different features across multiple classifiers. This IGene score also allows us to determine the expression of a gene in a sample. One of the previous challenges of machine learning and artificial intelligence applications in precision medicine was that there was no way to do a differential expression analysis. Differential expression analyses are really important as they are how enrichment is determined in cross-condition samples. You need to know which biomarkers are upregulated in a case or a target sample and which ones are downregulated to get good results back on an enrichment analysis. IGene scores are the first way to do this with a machine learning application. We'll dive a bit more into those in just a second, but I want to first talk about how these IGene scores can be used for looking at the relationship between a biomarker and a disease. IGene scores for each of the features in the feature set that you use to train a model will be scored from 0 to 1, with 1 being the most relevant. Each score is then ranked according to their relevance to a certain predictive condition. Essentially, the biomarkers with the highest IGene score were the most useful for making predictions across multiple classifiers. This usefulness usually indicates some sort of biological relevance, which we'll demonstrate when we go into the heart failure case study. I know that was probably very abstract, so let's look at an example here. So here we have an experiment where a user used 3 different predictive algorithms within IntelliGenes. A random forest, a support vector machine, and an extreme gradient boosting predictor. One biomarker was chosen as a useful biomarker for predicting a disease, and SHAP importances were generated for its predictions. As you can see, judging by the blue color here on the low SHAP values, downregulated genes, downregulation of this gene was very useful in predicting the control of this experiment, which is, again, in this case, non-heart failure patients. A negative SHAP value implies that you are efficiently predicting control, and a positive SHAP value implies that you are predicting a case with high accuracy. The shading on these dots here is relative to the level of expression as in relation to that data set. So these blue dots here represent underexpression, and these red dots represent overexpression. We see that these results are consistent throughout each of the 3 classifiers. So what we do is we score this specific feature as underexpressed in controls. The reason it's scored relative to controls is because controls has a bit of a higher SHAP score between these 3 classifiers. It's very hard to notice in these figures, but trust me, it is there. So once again, with these SHAP scores, we are compiling feature importances across different algorithms. We are determining the regulatory status, so upregulation or downregulation, without performing a differential expression analysis. And then we're ranking biomarkers based on their usefulness in predicting classes, which usually translates to some type of biological relevance. So with that all being said, I want to first introduce this scientific reports paper, which is one of the most cited papers in precision medicine this year, and then bring you through some of the visualizations that we can create with IntelliGenes that we created in this study. One of the big selling points of IntelliGenes for us is that it has a wide suite of visualizations. We think that interpreting this type of data is really important for scientists who might not have the technical background, because it very easily contextualizes what is happening within these predictive models. So for this study, we collected RNA sequencing data from a very small but diverse cohort of patients. We utilized IntelliGenes to create a predictive model, which we've published in this study. And we were able to determine if a patient was positive or negative for heart failure with a 91% accuracy. So this model performed incredibly well, very highly discriminatory. Additionally, we did some downstream investigations from the data that we derived from IntelliGenes. We examined the biological pathways that certain biomarkers fell into, as well as the networks of genes and diseases that were created from this kind of set of 18 biomarkers that we had. So the first plot that we generate in this study is a correlation matrix. 18 features, as I said previously, were selected within the study. Those are the 18 features shown on the X and Y axis. From those 18 features, we were able to create a gene-gene correlation matrix. What we found is that when we use this correlation matrix with IgG scores, we get back very interesting results. Genes that were highly correlated tended to have similar IgG scores, meaning that they were biologically redundant in a way, but served as a backup mechanism for our predictive algorithm. The correlations between biomarkers could also be examined further by looking at the regulation status through an IgG score. If we saw that two biomarkers were very highly correlated, we could examine their relationship. Were both biomarkers upregulated within similar patterns? When one biomarker increased, did the other decrease? Things like this were directly able to be gathered from the IntelliGenes platform, which would usually require, as I've said previously, a differential expression analysis and then more downstream analysis. We used IntelliGenes to reveal gene disease networks. Labeled in green are those 18 biomarkers that we found. In blue, we have ICD codes for diseases that are not related to CVD, and in red, we have CVD diseases. What we found is that the 18 biomarkers in our study that were derived from IntelliGenes were very densely connected. If you look at this marker here, for example, EGLN2, you can see that it is surrounded by other genes from the study and links to numerous diseases that we either found to be risk factors for heart failure and cardiovascular diseases or CVDs themselves. There was only one gene that was not connected to this network of the 18 others. That was this gene up here, TWF2. We also discovered that the majority of the genes that IntelliGenes found unbiasedly had a direct correlation to CVD previously reported in literature. This is an additional validation that our selection algorithm is working as intended. Because IntelliGenes is a non-biased platform, meaning it is not reliant on previous annotation databases, it can be applied to any disease or condition. If you have data for a certain disease or condition you are interested in studying, you can use IntelliGenes without doing any prior literature search and generate a list of markers that we are confident are disease associated. This is the confusion matrix that we generate with IntelliGenes. This is a scoring of essentially what the predictions that the algorithm is making fall. Here we had 22 patients in our testing group, or in our in our testing group. Sorry about that. We predicted 17 of them as true positives and 3 as true negatives. This shows that our model is not only capable of predicting the disease class, but is also capable of predicting controls. This is quite an unbalanced data set. We have more CVD patients than we do have healthy controls, but IntelliGenes was still able to produce a high-performing model. Additionally, we saw a single type 1 error and a single type 2 error, meaning that the predictive models aren't skewing more towards one of the classes rather than the other. When I say class, just to be clear, I am talking about a case or control subgroup. But yes, this is the 91 prediction accuracy that I had presented. In upcoming studies, we are actually using this model to predict on a completely separate data set, meaning that the models that IntelliGenes produces are not only accurate, but they're generalizable. If you train a model on a set of 100 patients, you could use it on a completely unrelated set of 100 patients with the proper normalization. And then the final thing that I'll go over today is the feature importances and the linkages that we see between different features across algorithms. So here I've pointed out an ensemble ID ending in 618. This was a feature that we saw as incredibly important across multiple different classifiers in the prediction mechanisms that we ran. When we're seeing a feature like this, that is the highest scored feature in XGBoost and k-nearest neighbors, two very different machine learning algorithms, we can be quite certain that this is a biologically relevant feature. Because we can compare so many different machine learning algorithms within the same suite, with IntelliGenes, we find plenty of these markers that are performing well, no matter the computational background of the model that we've trained. We really want to emphasize these markers going forwards. We think the IGN score does a really good job at doing that. And we believe that these markers are the most important when we go back to study some of our computationally predicted markers in an experimental setting. With that, I am going to hand it over to Rishabh, who will be giving you a demo of the platform. This is the interactive version of IntelliGenes. This is the graphical user interface that is incredibly user-friendly. If you do want to join along in the demonstration, we hope that you do. We're here to answer any questions that you have. And with that said, I will hand it over to Rishabh.
Thank you all for the incredible explanation of the IntelliGenes platform. And of course, thank you for joining our workshop. I'm Rishabh, the primary developer and author of the interactive IntelliGenes platform. I will also share my screen so that I can go through the presentation.
I believe mine is stopping right now.
Yes. Are you able to see my screen? Well, can you see my screen?
I am getting an issue with the page being unresponsive. Give me 1 second.
I can see your screen. You're good.
All right, perfect. Thank
Now,
you.
yes, I can.
All right, great. So hey everyone, I'm Rishabh. Like I said, the primary developer and author of Interactive IntelliGenes, the graphical user interface behind our IntelliGenes methodology. But to get started, before we get into the demo, I'd like to give a bit of a background into the motivation behind building such a tool. So IntelliGenes, just to recap, as Will already described, was designed as this robust AI/ML framework, which is effective not only at disease prediction and biomarker discovery, but it uses like an ensemble, a combination of different machine learning algorithms and statistical models. Our second version, our interactive version of IntelliGenes here, was created to address some of the limitations of the CLI tool. Specifically, we were focused on improving the general accessibility of IntelliGenes, making it more accessible to the wider scientific community, as well as to improve upon the existing visualization and interpretation capabilities of the framework that Will had described. So one of the drawbacks of the CLI version that we'd experienced was that users still required some basic understanding of, or some basic computational experience. For example, understanding of how to navigate the command line, or executing basic Python scripts, or understanding and handling file paths. All of these skills serve as potential barriers to entry, and are some skills that many researchers and clinicians in industry today lack. Specifically, when IntelliGenes CLI was actually demoed to a classroom at Rutgers University, we noticed a problem that many students who lacked this computational background sometimes would struggle with the installation or the usage of this tool. We saw that there were many parallels between these students in classrooms and potential scientists in industry who'd want access to this more complex and effective machine learning prediction models. And we realized that in order to make these types of analyses more available to the wider scientific community, regardless of prior experience, this challenge needed to be addressed, right? And we hoped to address this limitation, or we aimed to address this limitation by creating this more accessible version of IntelliGenes, one that could be used easily without any computational background or understanding of things like the command line. Specifically, we developed a graphical user interface, which we refer to as a GUI or a GUI. I might use these terms interchangeably, but they mean the same thing. We also published this article in the Oxford Academic Journal, Biology Methods and Protocols, which as you see on the screen. So first, to give a general overview of the tool, Will did a fantastic job explaining the architecture itself, so I won't go too much into the methodology or the reasoning behind the specific algorithms. But at a high level, the interactive version of IntelliGenes was built to make this robust analytic workflow that IntelliGenes introduced more accessible to scientists that may lack the computational background. But I want to stress this, that although the primary audience are these non-computational scientists or clinicians and researchers without this background, even those researchers with the computational background, such as myself or many of you, still can benefit from the overall streamlined and user-friendly workflow that the interactive IntelliGenes offers. And so at a high level, interactive IntelliGenes is composed of 3 different stages that mirror the general data processing workflow. And these 3 stages are data manager, AI/ML analysis, and visualization. Data manager was a section that is, I'll explain each of these sections in further detail in a couple of slides. But when building the application in each of these stages, we set out to achieve a set of goals, right? Because accessibility was the forefront of building this application. It was the reason why we wanted to make such an intuitive platform. We wanted each section to be very simple and intuitive to use, but to still be highly configurable and allow for the same kind of complex and powerful analysis that was offered by the CLI tool, by IntelliGenes as a platform. Moreover, we wanted to stress that the CLI and the GUI tools were cohesive and part of the same underlying methodology with the same algorithms and efficiency and efficacy. So to go over each of the sections in a bit more detail, I'll first talk about the data manager section. So the data management section is where the user can configure their input, right? So as Will mentioned, IntelliGenes works on a CIGT formatted dataset, which is a dataset that allows the integration of multiomics data, genomics data, with clinical information about the patient, such as their age, race, and sex. During this phase, the user can select and modify their input data set. They can tweak their CIGT formatted data set directly within the interface, as well as configure all necessary information about where they want to save their results to. During the demonstration, I will walk through the platform in more detail so that we can see exactly how the different components interact with each other. Here is a screenshot of the data manager section. At the very top, we have an option where the user can select an input file. This allows them to navigate their file system and upload a CIGT formatted dataset. Moreover, for added convenience, they can directly inspect the dataset within their platform or within the application, and then moreover do some minor edits and tweaks. For example, deleting specific columns that they decide aren't relevant or specific rows. They can do all of that within the dataset. Moreover, after they make changes, they can save their changes as a new dataset. They can revert any changes that they didn't mean to do. And overall, we designed this application to be very reminiscent of a basic spreadsheet editing tool, like you would use Excel. We wanted this to be very accessible to all scientists who would use this platform. And so we make zero assumptions about their prior background experience. They can select after they're configuring their input where they want to save all the results of subsequent analyses. And this is just, again, to make sure that it's their analysis is organized and easy to work with without having to do anything on the command line. Our second stage of IntelliGenes GUI is AI/ML analysis. Like Will said, we have an ensemble of different machine learning algorithms and statistical models. And here's where the user can do all the same configuration and tailoring of their analysis for their specific data set. What we wanted to emphasize with our interactive version of IntelliGenes is that it is consistent with our CLI tool in the sense that the results, as well as all the configuration options, are perfectly identical between the two tools. This is to ensure that the bioinformaticians using the command line tool or potentially the non-computational scientist using the GUI tool both have a similar experience and are both able to perform the same powerful analysis without compromising on any feature sets. Will already did a fantastic job explaining the architecture of IntelliGenes, so I won't cover it again. But at a high level, IntelliGenes, again, has a dual phase analysis where first we do a selection algorithm where we select relevant features in predicting the disease state. And then finally, an ensemble of machine learning classifiers that are used to actually predict the disease state, which we aggregate together with a voting classifier. Finally, what we wanted, or what we noticed when developing the IntelliGenes graphical user interface, is that it's very simple with the CLI tool to track progress and how execution is going, because you can just see the log statements in the terminal. With the GUI, we didn't want to compromise on the same convenience, and so we implemented this execution console, if you will, where the user can track progress and debug any potential errors that they might encounter in the same way that they would with the CLI tool. During analysis, we generate numerous different metrics and visualizations, which will be saved to that folder that they generate or that they selected in their data manager section. So it's very organized. First, they deal with input, then they deal with analysis, and finally, they'll deal with visualizations. Here, again, we see a screenshot of the analysis tab. At the very top, the user is given the option to select the pipeline that they want to execute. So we have support for, like I said, feature selection, feature classification, and then just for convenience, a pipeline where they can do both at once. Below each pipeline is a set of parameters that the user can configure using buttons and drop-down menus in order to easily tailor their analysis for their specific data set. What we wanted to stress was that the IntelliGenes AI/ML analysis tab provides the same exact compatibility with the CLI tool. So it's very easy to get started. It's very easy to interact and select specific classifiers. For example, they can change specific, they can disable specific classifiers that they aren't relevant for their data, or they can change the significance thresholds to make it slightly stricter or slightly looser, and so forth. Once the tailored parameters are selected, the user can execute analysis and, like I said, track the progress of the execution directly within the console. Here they can monitor the progress and also see if any errors or any unexpected results occur. And lastly, the last tab that IntelliGenes, or the last module, is the visualization stage. In this stage, the user can directly inspect all the textual and graphical results that are generated during analysis. And we validated that the results generated by both the graphical user interface and the command line tool are fully consistent, which again is just to reinforce this idea that it's all under the same umbrella of like this IntelliGenes framework. Moreover, what we wanted to stress when developing the IntelliGenes interactive version was to empower the users with more useful visualizations and charts. So the interactive IntelliGenes generates further visualizations such as ROC curves, confusion matrices, more plots about the correlation and distribution of features. And moreover, in addition to generating these plots, it allows the user to directly interpret and analyze these data sets within the editor itself. So here we see the screenshot of the visualization tab. First, at the very top, the user is presented with a list of all the results, which the user can select and inspect in this little screen at the bottom. They can navigate images, zoom in, zoom out, just to get more detail. And they can also filter the results for more convenience if they want to select or inspect very specific visualizations. So here's, for example, some of the visualizations that we generate on top of the command line interface. So Will already described the SHAP scores at the top, but here, and the confusion matrix. But to give a brief overview again of the different plots, here at the top left, we see a confusion matrix, and this gives us a good insight to the general accuracy of each model by comparing the predictions with the true values. So here we see that for this particular configuration, the model performed fairly well, getting and is fairly high at discriminating, only making one error here. Below that, we see a pairwise feature correlation plot. So for example, for all the selected significant features, we plot their relationship with other features. And this is useful in determining how these different features are related and correlated together. On the top right, Will already described the SHAP scores, which essentially just measure how good is each feature in, or in what direction does each feature affect the prediction, and it can be useful in figuring out, again, the underexpression or overexpression of specific data points. Below that, we see both the feature correlation plot and the ROC curves. The correlation plots, similar to the pairwise plots that I mentioned, help us understand, again, how strongly correlated the features are with each other, whether when one feature increases, the other one is likely to decrease, information like that. And the ROC curve, if you haven't had experience with it before, is a measure of the true positive rate versus the false positive rate. So it essentially measures whether a model is good at discriminating the data. Specifically, a curve that's closer to the top left corner is better at discriminating, whereas a curve that's straighter, more diagonal, is typically a poorer model at predicting the different conditions. And lastly, at the bottom, we have this distribution plot, which here is a box plot that gives us useful information about the distribution of features, as well as information about specific outliers and things like this. So overall, each of these plots provides the user with some additional information that they can use to interpret the results and understand their underlying data set. They can determine information about the efficacy of specific models, and it can also help them guide further downstream analytic decisions. Okay, here after we've briefly given an overview of the IntelliGenes platform, we'll go through a little demonstration of how to use the tool, installing it, and running it on a sample data set. But before we do that, are there any questions about IntelliGenes, whether the CLI, the methodology in general, or the interactive IntelliGenes and the visualizations that we generate with it? If there are any questions before we get into the demonstration, we'll try to answer that now, and then we'll go into talking about the demonstration. I will also share my screen so that we can see my entire screen. Are there any questions? Okay, I see one question. Do we need a GPU for running the program? No. So IntelliGenes is fully capable of running on platforms with poor hardware system or poor hardware availability. So even if your system doesn't have a GPU, although it may take more time to execute, it's perfectly capable of running and executing on these lower hardware systems too. So no, a GPU is not required. And one thing to stress that both the CLI tool and the GUI tool are fully compatible on all the major operating systems. So it should work whether you're running Windows, Mac OS, or Linux. All these platforms will work just as well the same. Okay. I will share my screen for the demonstration. If there are any more questions, Will obviously just let me know, and then I can try to answer any of those questions. All right, so I will share my screen and we can get started on the demonstration. So first we'll demonstrate the graphical user interface and then Will, I believe, demonstrate the command line interface as well. So here we have the best way to get started with the graphical user interface is to visit our GitHub and download one of the pre-built releases for your operating system. So the link to the GitHub is here. Will, if you could paste that into the chat so that they can access it, that would be great.
Absolutely.
Well, once you go to the GitHub repository, you should see a releases tab here where you can press the link and then download a specific executable for your operating system. So like I said, we support all the major operating systems, including macOS, both the newer silicon models or the older Apple Intel models. We also support Ubuntu Linux and Windows x86. So if you're running any of these platforms, you can download the pre-built releases. Unfortunately, if you're not running one of these platforms, there's no pre-built release, but you can still download the repository and build it from source. If you would like to do that regardless, we have a YouTube video here, which guides you through all the steps needed to build the application from source. But like I said, the easiest way is if you're running one of these platforms, just download the pre-built executable and then install it on your computer like you would any normal application. So for example, here in the GitHub page, you can go to this releases tab here, which will take you to this page and you can download. For example, for my computer, I have this, I'm running an Apple Silicon computer, so I would download this first one. Once you download the actual executable itself, the application, we recommend that you download one of our sample data sets. So here, if you go to the tests folder on the GitHub, we have two data sets, this dataset.csv and this counts CIGT CSV. Both of these are CIGT formatted files. This dataset.csv is the one I'll use for the demonstration today. It's slightly smaller, but it's easier to interpret and understand right off the bat. So you can download this dataset, and to do that, you can press it, and GitHub allows you to download a file directly. I have already done this step, downloading both the application and the dataset, but make sure you download both so that you can run this demonstration. All right, so here I have this file already downloaded, both the IntelliGenes application and the dataset in my downloads folder. What we could do is we can open the application. When you first install it, the setup might take a second or two to install, or a few seconds, but give it some time. And then once it's opened, you should be presented with this page. It opens directly to the data manager section so that you can configure your input file. I'll give everyone a few seconds or a few minutes to try to install the application and to open it up and make sure everything's working. If you're having any problems with the installation of the pre-built executables, then please let us know. We'll try to debug the error afterwards. Or if you're willing to try building it from source, if you have that computational background, then you can give that a shot as well. But I'll give you a few minutes for everyone to catch up to me right here, and then we'll get started with the demonstration. And while everyone's doing that, just to give a brief overview again, we have the Data Manager tab where the user can select and configure their input and output files. We have this AIML analysis tab where the user can configure their the different parameters that will be used during analysis, and they can actually execute the analysis. And then they have the visualization tab, which will allow them to inspect specific files to preview. Okay. So once you have this application open and the Data Manager tab selected, you can go and navigate and select the actual input file that you downloaded. So I'll just navigate to my downloads folder and select this CIGT formatted dataset that we have here. So this was the one I downloaded from GitHub. Just look and search on your computer for wherever you saved it, and then load that into the application. Once you do that, it should automatically fill out this table, which contains all the information that was present in that dataset.csv file. So here again, like Will mentioned, we have two columns, that the type and the ID, which represent, or the ID column, which represents just a unique marker for each patient. The type, in this case, 0 being a control patient and 1 being the case patient. And then a series of genes or features that are used during the predictive analysis. So here, after, you can make specific changes. For example, let's say I wanted to delete these two columns because they weren't important to my analysis. I can remove them, and now you see that they're gone. If I realize that I didn't want to do that change, I can reset that, and it brings the data set back to its 0 state. So this, again, is just to make very minor edits. It should feel reminiscent of like an Excel or Google Spreadsheets type software, just to be easy to use and make minor edits before doing the actual AI/ML analysis. After that, the user can select a specific folder that they want to save it to. By default, it'll save to the same folder where their input file is, but I typically like creating a separate folder just to organize the results separately from the input. So I'm going to create that folder on my computer and open it. You can follow along how you would on your computer with the graphical interface. And at the bottom, if I wanted to, I could reset IntelliGenes, which essentially just clears everything and brings it back to its original state as if you had just opened it. And this is useful if you just want to start from scratch. You realized maybe you loaded the wrong data set or something, and you just want to reset the application. So once the input is loaded, you've made all the necessary edits, and you've selected your output location. You're then guided to the second tab, which is AI/ML analysis. And this is where you can select a specific pipeline. For example, feature selection, which will just select, using the statistical models, which features are significant towards predicting the disease state. Here you can configure, again, information like the percentile for recursive feature elimination, or the alpha significance thresholds for each of the statistical models. And you can also configure things like the random state, which is useful for the reproducibility of results, which is important in the scientific community, as well as increasing or decreasing test size. We have all sensible defaults. These are typically accepted values in the scientific community, with the alpha values of 0.5 or 5%, and then a test size of roughly 25 to 30%. Feature classification and a combined method to run both. Here I'll run this basic or the first selection and classification one, and I'll just leave all the default parameters. Of course, you can go in and tweak specific ones that you for your specific analysis. But here I'll run with all the defaults. And when developing IntelliGenes, we made sure that we kept sensible defaults so that opening and running analysis right out the bat should work fairly well. Obviously, you can tune these parameters for your specific analyses to get better performance and better results. So once I decided that all my parameters are set, I can execute the analysis. And the moment I do this, it'll start executing and then logging all the progress within the console. For this data set, since it was fairly small, it finished super quickly, and you get a message at the bottom saying that it finished. For larger data sets, this may take more or less time, right? So, but this progress allows you to visually see that changes are being made and that progress is being made, and as well as debugging any potential errors or unexpected results you might face. You can be done through this interactive console. Lastly, once you've executed analysis, you can head over to the visualization tab, wherein all the results that were generated from analysis are automatically populated within this list. You can go and select any specific plot and then preview it. For example, let's say I open the confusion matrix for the random forest. Once I open an image, it will render the image in here. I can scale it to fit into the window, or I can zoom in if I want higher detail in specific sections and scroll. But I'll fit to window here. Again, this allows you to inspect the different results that were generated. And we already discussed some of the benefits of having numerous machine learning models. For example, here we can inspect both the for the random forest or the k-nearest neighbors. The different models perform differently, and you can analyze the differences between each one by looking at the different results generated. You can look at, for example, let's say, the correlation plots of the features, or some of the ROC curves, the SHAP scores for specific models. Textual results that are saved will be shown in this grid-like format, easy to inspect. Here we have the SHAP scores that we discussed before. Essentially, this is just a way for the users to inspect all the results and try to actually interpret them. On the side here, because this list gets fairly long and can be difficult finding exactly what you're looking for, we provide just a simple interface where they can select specific values. Let's say they want to look at just the distribution of specific plots, then they can select a filter and look at just those results. So again, these are just, this interface is meant to be very intuitive and accessible to the wider scientific community because it doesn't require any computational background. We strongly believe that providing access to these types of complex analyses for scientists, even without prior experience, is important because it opens up the door for discovery and analysis to more people, which is always a good thing. So this is the IntelliGenes platform. I hope you were able to install it and use it on your computer as well. Follow along with the demonstration. I will turn it over to Will for the command line tool. But if you have any questions about the graphical user interface, if you're facing any errors, I'd love to talk with you about it afterwards, after the end of the presentation, and we can maybe debug some of those errors you might be facing. But this was the graphical user interface. The goal being to make this sort of analysis highly accessible to non-computational scientists as well. Okay, I will turn it over to Will. And if you guys have any questions, please let us know in the chat, and then we can resolve that afterwards.
Yeah, Rishabh did cover all of the features of the platform itself. I'm just going to go through the command line implementation of IntelliGenes very quickly, show you where to download it and how to do so. So let me share my screen, and we'll go over that now. Is this visible?
Yes, it is.
Awesome. Thank you. Okay. So if you are interested in using the command line implementation of IntelliGenes, you can visit the GitHub main page for IntelliGenes, which has all of the instructions you might need right on there. So I have already preloaded an environment that has Python installed. We are using, I believe it's Python version 3.10 today. If you scroll down on the main page of this GitHub, you'll see the instructions on how to utilize the platform in a CLI environment. A CLI environment can be very useful if you are running very large jobs that might be time intensive, although that would require a lot of features and or samples for the platform to require such hardware. So if you do want to download IntelliGenes on your command line, whether it's a Slurm system or a personal computer, here's how to do so. You can simply copy the code from the GitHub. So we can clone the repository. We can cd into the repository, enter the directory, and we can use the pip install command to install IntelliGenes. Once it's installed, we have test datasets available in the repository already. So for example, today we'll look at this CIGT file, which is, I believe, 20 samples. This is the same file that Rishabh showed the results for. So what we do is we do iGenes, we navigate to our input file. This is in tests, excuse me. We create a results directory. We'll just create one right here. And we can run the platform with a simple command. Again, this is a 20-patient data set, and with IntelliGenes, it finishes in about 30 seconds. So let's navigate to that results directory and see what we have. From the results directory, we have the selected features, the visualizations, the classifier metrics, and the IGene scores, as already showed off by Rishabh. If you are interested in using the CLI implementation, this is how you do so. And I believe with that, we will turn it over to Dr. Ahmed to close us out. If there are any questions about either of the versions, feel free to ask. Thank you for being here with us today. I hope you were able to learn about IntelliGenes and maybe execute the platform. Additionally, I will put our emails on screen in just a minute in case you have any questions afterward to reach out. Thank you.
So with that, I think William and Rishabh, you both have done a great job. Thank you so much. And I'm also grateful to all the participants who are here. Please let us know if you have any questions. We are here for next 5 minutes and more. And if there is no question, then we definitely would love to close the session. Yes, you're welcome. And Will or Rishabh, any of you, please put the link to the lab and to the manuscripts in the chat box. Yeah. So in any case, they need to contact you or Rishabh or lab, they can feel free to do that.
Of course.
And I would add some final notes, and that is like we are continuously working on this project, and we are looking forward to come up with the new version of IntelliGenes in the near future, where we are coming up with more improved and better visualization and interactive interface for the global community.